So welcome everyone to session 4. We're going to talk about annotation, identification, and enumeration approaches. So first up is Joey O'Connor. He's going to be talking about identification approaches. Joey is a marine survey manager at JNCC. He manages the planning and delivery of JNCC's offshore seabed surveys and their outputs. And he's recently taken on the role of the NMBA QC Epibiota contract manager. And next up, originally John Hawes was supposed to speak today, but unfortunately he couldn't make it. So thankfully Henk van Rein and Ross Bullimore have graciously stepped in to speak about enumeration approaches. Henk is a marine ecologist working for JNCC. He's been designing, conducting, and analyzing benthic monitoring surveys in offshore MPAs for the last 5 years. And Ross is a marine monitoring program scientist at CFAS, and he supports the ongoing development and implementation of marine biodiversity programs. And lastly, Mark Burton will speak about the annotation software. And Mark is a marine scientist with Natural Resources Wales. He's been working at the SCOMER MCZ for the last 20 years, working on a wide range of marine monitoring projects, most of which involve imagery data. So I will pass it on to Joey to get us started.
Okay, so I'm going to talk to you about identification approaches used for benthic imagery. I'll give a little bit of background. I'll talk quite a bit about the ID Approaches Project Working Group, and then yeah, hopefully we'll have a bit of discussion either directly afterwards or at the end of all of the discussions in this session. Okay, so in terms of the context, in terms of idea of what I'm generally meaning identification of all of the living things that we can see in the benthic imagery that we collect, that we can see, and that we know, what the thing is. What kind of approaches? So thinking about the kind of Linnaean taxonomic approaches, species, genus, etc., but also for imagery, morphological approaches such as the Taxonomic classification, where we're looking at what a thing looks like, which can be a lot more relevant to some species than others, especially when it's quite hard to get to species level with imagery. And also thinking about a combined approach where we're looking at having in annotation software, for example, a label tree where you could go to your species level, where you can using your taxonomic approach, but also describe morphologically and apply that approach to your imagery annotation. In terms of the requirements for good identification, I think it's generally important that your ID is accurate and precise. And I know we spoke a bit this morning about that with Kerry, and maybe some of the limitations around individual observers and subjectivity between analysts, and even between the same analyst. So being accurate and being precise is important generally, but is very, very important when you're then looking to use the results of that ID to make comparisons across time or space. Say, for example, for monitoring change in a epifaunal community. It's also good if your identification approach is image quality appropriate, because we get quite a large range of image qualities across the different types of imagery collected throughout this group. Again, this is generally important and very, very important when it comes to making comparisons between data. That's a kind of context. So I'll now talk a bit about the well, lots of acronyms, but starting with the BPIDA PWG, which for those of you that don't know is the big picture identification approaches used for benthic imagery project working group. The group was formed through a workshop held by the big picture group back in November, which a lot of people at this workshop will have been involved in. And the idea of that workshop was to bring together people to carry forward tasks that have been identified in a benthic imagery action plan that Henk would have talked about yesterday morning. And to do that, we grouped the tasks into what seemed like sensible kind of themes, had some breakout groups to discuss those themes, see if they were sensible, see what bits of work were seems being most important to take forward, and then project working groups were formed from that. One of which was the this Identification Approaches project working group, where people in the group to form the group agreed that the main issue to tackle is around identifying the most appropriate / maximum level of taxonomic resolution and best classification scheme for the specific questions you're asking. So it's quite a big issue. And in November, people in that group agreed that there could be two kind of work streams to try to resolve that issue. So one focusing on the taxonomic or standard open nomenclature identification, the other focusing more on the morphological identifications, for example, through Katami or an equivalent. The tasks from the benthic imagery action plan that the group considered relevant to the group. The key one is this task 43 around recommending minimum requirements for taxonomic and morphological identification, again to meet different benthic imagery purposes. And the comments around what should be done to do that focused a lot on delivering guidance, something like a flowchart combining the various purposes, the various image qualities that people could then use to arrive at a recommended maximum level of taxonomic resolution for ID, which could then be applied consistently by people. Other tasks that were considered relevant from the action plan are the task around the EIP, which we talked about yesterday, the Epiphone Identification Protocol, and also a task around piloting a combined use of a morphology-based classification system with the EIP. Other tasks that were considered to kind of fall under the remit of the group are task 34 and 35. So 34 is to review taxonomic identification approaches for benthic imagery, and 35 is around minimum size of organisms that should be counted. And then in terms of that November workshop, there were representatives from these organizations present. So according to the workshop, in terms of who was doing what, my name down as the project working group lead, and hence I'm delivering this update. And Natural England and Natural Resources Wales participants said they would be able to collaborate into the future. And at that time as well, if people were told that Tammy Horton at NOC, who I believe is on the call, is doing work currently linking to the EIP deliverable. Some of that work is in this paper, which was mentioned yesterday. I have it on good authority that it's a very good read. So please do have a look at that if you haven't already. Next thing that we did was we decided to have a scoping meeting for people who had expressed an interest in the group in back in November. So we had that on the 12th of February, so quite recently. We used our time there to consider to look back on how the group was formed and to try to use that information that I've just sped you through there to I guess get a better grip on the remit of the group. We had a general discussion with everyone who could make it. We asked these questions. So what I'll do now for the next couple of slides is talk you through some of the responses from people, which will hopefully give you a feel for the group membership and what people are wanting to do over the next year or so. So with a wide range of people in that scoping meeting with a wide range of interests, all linked to image identification. So quite a few people were already involved in big picture work through the quality assurance framework and the epifaunal identification protocol. There are also people who had been participating or are participating in some of the other project working groups. There were people present who do a lot of contracted analyses. So, for example, from MPA monitoring surveys, and apply identification approaches doing that. Other work being done by group members include developing OTUs both for deep and shallow waters and through the Smart Ready framework that Kerry mentioned this morning. And online annotation was a bit of a common theme for people on the group. In terms of what we wanted to do next year, we came up with quite a long list. So further developing the EIP and the quality assurance framework was seen as a priority for people. Kind of going back to the like some of the ideas from the November workshop. People like the idea of producing a flow chart which would combine purposes and image quality to recommend the maximum level of taxonomic resolution for identification. So that kind of guidance. I guess the need for an identification system which works well both for shallow and deep habitats was flagged. And something we came back to a couple of times was the need to develop standardized workflow processes for identifying fauna from imagery. And I guess linked to that, improving the quality and consistency of contract requirements. We also talked a bit about work that will be happening in other project working groups, which is quite heavily linked to the remit of the identification approaches group. So thinking about ways how we could help some of the other groups. So for example, mobilizing existing imagery data for the data flows and data archives and catalogs, project working groups. And ensuring that the work was quite well aligned, particularly with the enumeration group. Another thing that we identified as something to do was to document emerging imagery methods used within marine protected areas. And then I guess these final two are two of the extremes. So one, developing a UK specific system based on Katami. If no one else will or is doing that, I think that would be quite a big piece of work for us to take on. So it would be quite a good thing to aim to make some progress on over the next while. Perhaps more, I guess, readily achievable is this idea of collating a list of contactable imagery experts to share with the wider big picture community and maybe the community more widely than that. So this kind of comes from the EIP mini workshops that would have happened before Christmas and up to last month. So that kind of group where people could get involved with that community if they had specific questions on ID. We also agreed a group focus. So what we will do is build on the EIP work due to be completed later this month. And by building on that, we can produce guidance on the use of imagery to make formal taxonomic identifications and morphological identifications. And then we spent kind of the tail end of the scoping meeting trying to figure out how we could actually get the work done and funded. So on the funding side, JNCC are actively seeking funding to take the work of this group forward. And as you might imagine, the level of work that we can do into next year will depend on how this goes. Both we're very open to, I'd really like to hear any suggestions others have in the workshop on other funding routes that we could pursue, because as you have just seen, there's a huge amount of things that we'd like to do if we can. In terms of how much time people can contribute to the group, this is quite closely linked to the previous slide around the other resourcing. But what we agreed is that people would contribute the time that they had available. We'll have active members and passive members. It's quite likely that that will be fairly fluid, and people will be active when they can be, passive when they can't, vice versa. But the idea is that all group members will receive regular updates on progress against the things that we plan to do, as well as from the Action Plan Coordination Committee meetings, and that they will have the opportunity to input to and review the outputs of the project working group. The ties to other project working groups. There are 8 project working groups. One of them is the benthic ID Approaches group. The other 7 are listed here. I couldn't think of another group that is there that the ID Approaches work doesn't feed into. So it's maybe a bit of a redundant slide, but there you go. And as touched on, there is quite a I guess a stronger link with the Enumeration Approaches group even than some of the others, in that a lot of the, well, as we'll hear in the next talk, a lot of the work of that group is really closely linked to the deliverable around the most suitable ID approach and also the epiphone identification protocol work that we hope to build on to next year. One thing we failed to do was think of a snappier group name. So if you have been listening to this thinking, well, I've got an amazing name for that group, that I can't wait to share with everyone. Now is your chance. Please pop it in the chat. I'd be delighted. Anything that doesn't have Mac in the title would be brilliant, and we can consider that. So the next stage is we want to formalize our plan, turn it from our kind of thoughts of what would be really nice into something a bit more tangible. Very closely linked to that, we need to secure some funding, some resources, and we also want to finalize the project working group membership. So this is an opportunity for people on the call who've not been part of this work before to step forward and get involved, either by letting me know throughout the workshop or contacting the Big Picture the email address, and we can get you involved. Ideally, this would all happen over the next month. So we'd be looking at facing into April with a plan, some idea of funding and resources, and a finalized first list of members for the group. That's all from me. So I'll take any questions if people have them, and yeah, hand back over to Steph to lead us through.
Okay, great. Thanks, Joey.
Thank you.
Yeah, if anyone has any questions or any name suggestions, now's a good time to bring them forward.
I see a question in the chat there, Steph.
Oh, sorry. Okay, yeah, we have a question from Barbara. She says, how many people are currently part of this working group? And do you have an idea of how many of these have experience with taxonomic or morphological identification?
Thanks. I think I'm going to say around 10, but I will pull up the list as we move on through the session and get you a proper number. I guess there was maybe 20 or so people interested in the November workshop, and maybe 10 people available at fairly short notice to join the scoping meeting. Maybe another 2-3 people interested in the like the kind of being part of the more passive approach and receiving updates as opposed to joining the group meetings. People, so in terms of experience with taxonomic morphological identification, I think pretty much everyone in the group has, in the scope meeting anyway, has done some form of taxonomic or morphological or both identification. But Barbara, I'm happy if you want to ask a bit more detail, please do.
Hi, thanks, Joey. No, it's just that I'm just, I guess, wondering about the diversity of people involved, and just, because if you do have people with that experience, it's important, right? So I was just curious. Yeah. Thank you.
Yeah. So we had, I guess there's the representativity is quite close to this, like the wider representativity in the big picture group, and that there's people from people who do imagery analysis, like contracted work, so from environmental consultancies. There's some academics as part of the group, and there are people from the SNCBs, which are the statutory nature conservation bodies. So I guess the public bodies involved in nature conservation in the UK. But I will have a look through the group membership, and because I'm sure I've missed out on something, and put an update in the chat in terms of numbers and representativity.
Henk?
Yeah, just come hand up for a quick question, like a quick comment, really. More an observation from a general overview of all the groups in the big picture group. Something we haven't really sort of made really clear, but this session we're going to be chatting to the groups who are only just getting started. So it's quite an active time in these groups where they're putting ideas together. I think every group has maybe met. Some, in some cases probably more meetings are needed, and it's very early days, really. So it is quite an interesting time for these groups and to be a part of them. So I guess that's my one comment. And my second one is that if you caught any of the second session yesterday that covered the EIP, the Epifauna Identification Protocol, in many ways that is like the flagship product or the flagship output from this group. And from that, there's massive scope to sort of branch that out and to begin to link up with sort of, imagery catalogs, perhaps, and sort of other classification systems and all sorts of things. So there's some really exciting stuff ahead, I think. Yeah. Thanks, Joey.
Okay, great. Thanks, Henk. Does anyone else have any other questions for Joey or about the group or? Okay. Yeah, I'm not seeing any other comments or any hands up. So I think then we can move on to Henk and Ross.
Sure, thanks, Steph. Okay, Ross Bullimore and I are going to do a bit of a tag team thing that hasn't been rehearsed. We're covering for one of our colleagues who sadly can't make it today. So bear with us as we've cobbled together things here based on things that we don't really know. We haven't got the best information on exactly what's going on. John Hawes has been leading this work area, and he's got the best idea. What we do have, though, is some idea, and so I've put together just a few slides based on some of our past work and a bit of context on this project working group, and then I'm going to ask Ross at some point just to chip in. As you attended the first project working group meeting of this group, Ross. And so we're going to do a kind of a slide, a mid presentation sort of slide switch, I think, or something like that. I'm not quite sure how that's going to happen, and then round up on some sort of good conversation points, I hope. So just laying it out like that, I will bring up my screen here, and you can hopefully see this. Can you see that? Okay, great. Brilliant, thanks. Apologies, I haven't got Ross's name in here. I've literally just copied the slides from another session, changed the name, but we are talking about benthic imagery enumeration approaches. Or really, it's enumeration approaches of our targets, the things that we see in our images. Targets can be abiotic features, or they can be beasties, biological taxa on the seabed. What do we mean by this? So essentially, it's counting things of interest. Now, in some instances, when you're faced with an image, you don't really need to count things. You just need to record whether the thing you're interested in is there or not. So we call that presence-absence, right? In other instances, where you want a bit more information, where you want to understand sort of community dynamics, community ecology, it's important to know the amounts of things, or at least the relative amounts of things. So what you can see in the image here are, this is sort of a hard substratum environment, mostly sort of bedrock, and little patches of sediment that have sort of blown over it. And you have a few sort of solitary organisms, sort of brittle stars, scuttling about, and then you've got lots and lots of encrusting organisms that are sort of growing over there. Now, if you can count them, you can count everything, but really, is it technically appropriate to count colonial organisms? Probably not. So you can maybe estimate the percentage cover of them, and that's another thing that we do. In fact, often what has become standard operating practice for most of the conservation agencies in the UK is to do a split approach, where you count it if it moves, or if it's clearly solitary, and then estimate the percentage cover that it occupies in that entire image if the thing is quite solitary, especially if it's colonial, and you can't really count the number of individuals in that colony very easily from the imagery. So you end up with two methods really of enumeration for your one sort of survey. Another approach, which is more about relative abundances, is to use this system called SAC4. Bring up a little thing here. SAC4, so it's super abundant is the S, abundant, super abundant, abundant, common, frequent, occasional, rare. There are guidelines that have been published. Quite a few of you have been involved in these back in 2004. This is a long-standing system, sort of a rapid system for assessing the relative abundance of things, depending on the growth form, whether they are sort of a colonial thing, that grows in sort of meadows and crusts and so on, or whether it's an individual thing. And depending on the size or the percentage cover of the thing, you, the human being looking at the image, or if you're actually in situ looking at the seabed, you need to make a calculation in your mind of what it is. What could possibly go wrong with a system like that? So it is a great system. However, in our current sort of needs to monitor changes in seabed habitats, there are a few issues which I'll come back to. Another method quite common in the tropics, and indeed in many studies, is to throw some points across your image. And there are numerous studies where you can put a regular grid, as I have on this image, and count them. I think there are 100 points there, regularly spaced, or you could randomly organize them, depending on what type of data you're trying to get out of that, and on the distribution patterns of your organisms. Either one of those strategies might be more appropriate. There have been numerous studies comparing them, and many studies that say there's no difference. Other studies that say there is a difference. So, you know, that's up to you to decide on that approach, whether you want to do that. There are issues with it, though, in that it doesn't cover all of the image. You only get whatever's under the points. Another approach that we've recently been trialing, although it's not new, is to divide the image into smaller squares or grids, and to record the frequency of occurrence of organisms or target features within every grid cell. So you have 25 grids, 25 grid cells in this grid, and you could go around that image recording everything, the presence or absence of it in each one of those 25 cells. So if it's in every cell, it's 25 out of 25. That would be a high occurrence of that organism. Whereas if it's in just one cell, then it's only got 1 out of 25. And the advantage, what's quite cool about this approach, is that you don't have to qualify whether something is colonial or solitary. It just generates one data type for that image, which it can make the analysis a lot easier. So there's that. It could be a bit coarse doing 25 by 25. So yeah, you know, you could divide the grid even further into, you know, 10 by 10. And the same goes for the points. You can throw on more points if you want, or less points. And then it becomes a question of effort. And it is at the heart of enumeration approaches. You do have sort of a cost benefit consideration with everything that you do. In fact, with all of our work practices, really. But in this context, you want to use an approach to extract numeral information, sort of, you know, the counts of things. If that's what you're into, you want to get that information out as easily as possible, as consistently as possible, so that if you did it again, it would be a reliable thing. You could assess changes reliably in those things. Now, okay, that's sort of a rough overview, hopefully, of the kind of thing we're talking about with enumeration approaches. I'm not sure if all of you have tried all of those. There are probably other ways that you can get numerical information out of an image based on the beasties, but that's just an idea, though. Now, we raised this at the Big Picture Workshop a couple of years ago. And here we have Hayley Hinchin from JNCC. She's actually literally writing up what a group was talking about enumeration approaches at that moment. And there were lots of thoughts and ideas that went into it. And coming out of that, we obviously solidified those thoughts and ideas into the benthic imagery action plan. And the one section in particular, the top right, is all about imagery annotation approaches. Now, pausing just for a moment, below that, you've got the image annotation software and machine learning, or the AI stuff. Now, we touched on the AI this morning, and we touched on the data flows, or all the green stuff, earlier on as well today. Now, what we're largely talking about in this group, which I'm now going to go a bit more into, here are the tasks a bit more blowing up. Many of the approaches we're dealing with are immediate and day to day. The organizations that we've all been working with, these are things that have been ongoing in our workflows for 30, 40 years and more. But as time goes on, the more of this workflow will be carried out by AI algorithms, AI-based algorithms. And so in a way, we've got a transition from humans counting things on a screen to gradually using software to do that, to gradually a machine probably doing it all for us. So really, that's just to sort of get us to think about kind of the duration and timeline and things that need to change as time goes on. With these imagery annotation approaches, the three sort of broad groupings across this entire sort of theme. One, the taxonomic identification, Joey's touched on, which includes the morphology. And what we're interested in for this project working group are the enumeration approaches. There aren't that many tasks in here, but they're very important. They're at the crux of a lot of our quantification data from imagery. Yes, I'll just go back a second. So looking at these tasks, I mean, we see one crossed off already. This is a study that a few of us worked together on a number of years ago. I'll come back to that in a few moments. But other tasks such as reviewing approaches and exploring ways to improve consistency across approaches, deciding on the future uses of SAC4, developing minimum requirements for enumeration approaches to suit different benthic imagery purposes, and creating a decision tree to help people pick the right purposes. This idea of these are our standard benthic imagery purposes so far that the quality assurance framework is trialing. There are many others, but these are just the ones that are being trialed for the quality assurance framework. So for example, what we mean by that task that I just mentioned is that your feature verification, maybe that's got a nice suite of enumeration approaches that are best suited to just that. And that is just presence absence, actually. So in a way, that one's kind of done. But, you know, mapping, maybe the SAC4 method is good for that. Maybe some sort of frequency occurrence thing. Habitat species monitoring, maybe different approaches are best for that. There are other issues, of course, when you change the substratum type. You know, habitat types, maybe different approaches work better there. Different taxa, different approaches there. So it is very complicated. So although there aren't many tasks in this group, it will be quite challenging to drill down into this work. The work all focuses, and all of these different tasks, they all funnel down to this core task at the very bottom, which is basically about bringing everything we found together from taxonomic, morphological, and enumeration research into one place, and developing approaches that are bespoke to our needs. It's a bit of a general task, but everything's funneling down to that one point. So previous work, I'll rattle through this very quickly. We worked with mostly John Moore, you know, Alison Benson, Ian Sutherland, Tom Mercer, Matt Ferguson, and ASML and Envision to put together this report. Very early on in this process, we used different methods. The methods you saw on the slide at the start here. We looked at different metrics: power, precision, accuracy, efficiency, taxonomic richness, and consistency data. And we used those metrics to sort of compare these things to try and find out which method might be the best overall to go forward for a specific purpose. Our purpose, you don't need to look at most of this. Our purpose really was to come up with an approach that suited a high density, high diversity, temperate circulittoral bedrock reef community. This is a picture of where that was in Scotland, North Scotland. We had over 4,000 still images. We didn't have a lot of time on our hands, so we selected just 100 really good images, and we ran with those. And we gave those images to senior and junior analysts, looking to test consistency, that idea of consistency. And then we ran these 6 annotation approaches, the same approaches that I just mentioned at the start. Good working practice. We randomized the order of the images. We also, you know, explored UK current annotation protocols. This is pre-EIP days, and we recorded the time. There were issues pretty much, as John will tell you, pretty much as we got started, there were issues. So one of the first limitations were these methods have different data ranges. So percentage cover really is like 0 to 100, isn't it? And so one of the ranges of data is that. Another one of the ranges was abundance, was a large range. Your SAC4 has a very narrow range of data, as does the 5x5 frequency grid, and the others are more or less 0 to 100. So because of these different data ranges, you affect the variability of the data and the potential by which data can be variable, which will have knock-on consequences for your statistics, your analysis and your results. There were also inconsistencies in a taxonomic identification. I don't want to touch on those now. Joey's mentioned that already. But there were truncation issues where you sort of aggregate taxa together. There were those issues for the SAC4 and frequency data types. So that is something to bear in mind with these methods going forward. The key result, really, to summarize it all, this is a bar graph to summarize how things did, relatively speaking. Rank scores was the only way that we could compare things reasonably across these different data metrics, which are listed on the bottom, precision, power, efficiency, and so on. What you have is 0. 0 is the status quo. 0 is our standard approaches that we use in the conservation community, and that is using percent cover and abundance counts. So we use that for nearly all of our analyses. What you've got are 4 different colors here. Each one of these bars represents a different one of those methods of enumeration and how it performed using rank scoring relative to the status quo. So we see, if just look at precision, SAC4 does relatively better than what we currently do in terms of that, as does the frequency grid, as do both frequency grids, but the point data has a poorer level of precision. You see a similar effect with power. Everything's more powerful than doing what we were doing before. In terms of efficiency, only SAC4 and the small frequency grid were more efficient. In terms of taxonomic richness, everything was the same because they all covered the same amount of area of the image, apart from the point method. Do you remember the point? It misses most of the image. So there's a little bit missing there. In terms of taxonomic accumulation of taxa, well, that's the rate at which taxa sort of accumulate. They were all slightly better at that. In terms of community impression, now this is the sort of the relative accuracy of the data that came out of those other enumeration methods, and this is where all of those other methods, they perform quite a bit poorer. They create different sort of impressions of the community, sort of altered impressions of how the community looks in the data, not in reality, but in the data. And the consistency, important to know that the SAC4 approach was the worst for consistency-wise. You got the most variability between your observers. And the other approaches, you actually got lesser degrees of variability. So if what we're interested in is having consistent approaches, there's an argument here that these other approaches could be better to use. And then, of course, overall, everything summarized, you see that, yeah, pretty much everything performed better in these set of 100 images with these analysts and the approaches that we took, all apart from the point method. Now, that was a big surprise because we sort of thought that would do better. Yeah, the frequency grid came out as performing the best of all, but the take-home message there is that different people see the same things differently, and the different methods create different impressions of the same community. So it's all a bit of a mess. And what we would do better off is to come up with standardized approaches for standard purposes. At this point, I'm just going to skim across these. These are sort of ways of going forward, using EIPs, using annotation software, machine learning algorithms, standard classification systems like Smarter ID and Katami, having more precise targets and indicators, appropriate sample sizes. A big drawback of this study is that we were just treating images alone, and they are not samples. They are just images. They're almost a subsample of a sample. So this study would have to be done again. And I know others on this call have done this type of work really, really well. And we'd be really keen to hear from them. Obviously, we want to keep collaborating with the imagery community to try and make this better. So on that, we have our different project working groups, and Ross attended the Enumeration Approaches group. I'm sure they discussed this kind of stuff and lots more. So I'd just like to park myself there for a moment and hand over to Ross to hear about the kind of things that were discussed at that enumeration group, please.
Yeah, just before we do, Jen, do you want to jump in there?
Henk probably knows what I'm going to say.
I probably do, but others maybe don't.
Oh, just at the enumeration group, which we decided to rename the Quantification working group.
Ah, there you go. Good.
We, since not everything is individuals, as you mentioned, we talked about that report that you've just talked about. I just wanted to put out a caution that we shouldn't draw any conclusions from it for the reasons you mentioned. Basically, there's some fundamental issues with not using appropriate sample units and not having sufficient replicates. So I actually don't think we can compare the techniques in the way that's been shown. So that's something we're looking at in this quantification working group. But I just would caution people not to use that report as a reason for doing things one way or another, because I don't think the conclusions show what what it initially was thought it did.
Yeah. Great, thanks, Jen. That's quite a good point going forward. I think what we stumbled into was a hornet's nest there, and having more time and effort to focus that going forward, I think we'd be able to get even better estimates. And it sounds like that's what the group is thinking about going forward. Am I right?
Yeah, it's one thing we talked about. I'm sure Ross will talk about the rest of them. But pertaining to that study, yes, we did talk about potentially reevaluating it.
Brilliant. Great.
Brilliant. What I'll say before I start anything, Jen and others who were on the call with John week before last, feel free to just either stick a hand up or just jump in and interrupt me when I'm going off track. Right. Henk has pretty much done this already, but I will just whack this on the screen to give us some context. So the enumeration approaches, as Jen said, let's think in terms of quantification is point one, and that's quite a big change when we come on to one of the particular or two of the particular tasks under the group. As I said, John has been spearheading the project working group, and I am a terrible replacement for that, so I apologize. I've briefly started the list of people that I know had expressed interest in the group, and I think were definitely on the call the other week. I will have missed people. I apologize. It's just an indication, and others have been and will be involved as the work progresses. Henk's already put this up, but the little red box is roughly the place in the workflow where the focus of the group lies. But again, Henk's already covered this. It links in incredibly widely across the whole piece, particularly as Joey pointed out earlier on with some of the work from the identification group. And my last slide, and then I'll just cover a few bits. Just quickly, for anyone that has not seen the specific tasks from the action plan, or didn't quite get a chance to grasp the detail when Henk put them on the screen, I'll just leave these on the screen and just quickly run over a couple of bits from the meeting the other day, just from my notes. So I think the first thing to mention goes back to Jen's point about the kind of the agreement of the need to focus the group really on quantification, and how we do have this need to drive for quantification to inform what we're trying to do in terms of monitoring. So we're very focused on the right-hand side of Henk's table of imagery, purposes, and uses for the most part. But there's a couple of tasks in here which mention SAC4, and there is an agreement. I hope I haven't made it up. I think there is an agreement that there absolutely is a need to kind of take SAC4 to one side, and linked in with the bottom task in the plan about a decision tree of approaches and techniques to be used is being really honest and pragmatic and open about the right uses of tools and the right uses of the approaches. So it's very much a case of being really clear, open, and honest about what is appropriate to use and when. And certainly we did talk around this in the group, and there is a wide range of what SAC4 means to people. And one person's SAC4 should be, but is not necessarily the same as another person's SAC4. And we could go around that in much detail, Graham, to answer your little question that you put in the chat. And I'm not going to do that right now because I'll lose the plot. So moving on from SAC4, so let's get on to quantification. Something that comes up or came up and was discussed a bit, and certainly I might be going slightly off track of what we discussed in the working group, but just for folks' information, certainly within the kind of statutory monitoring SNCB DEFRA family group, something we're really trying to look at is in this kind of consistency of recording from different enumeration techniques, and how we can work with the legacy data that we have got and have collected over the past many years. What can we do with that? Because, without necessarily going back and reanalyzing. And a big thing about this is what Henk was showing in his slides a few moments ago. That difference between two different data types being recorded as standard. So this is the counts and the cover data set. So I can't remember how much it came up in something in the working group, but there's certainly something that we from within that group want to bring to the working group, is the techniques and the attempts that we're making at the moment to actually start looking at it's not a new thing at all, but to be combining the count and cover data, establishing what isn't appropriate, and how we are or are not influencing, a bit as Henk was saying, the impression of the community, by taking those two data types and bringing them together so that we can actually work with a single standard data set for analysis. So that's something that's actively going on in parts of the working group. Something else that we touched on, again, taking Jen's point absolutely on board with regards to some of the work that Henk was just sharing. Within the working group, based not just solely basing this on that report, but based on sort of the collective experience and the collective work going on throughout the range of organisations engaged in the working group already. I think I'm right in saying, I hope I'm right in saying that I certainly came away with the impression, I think the agreement was that what we're really aiming for and what we're really heading towards, especially in light of where we're moving with annotation softwares, is we think we're all heading in roughly the same direction, I think, around the cell count methodologies. But we've still got some way to go to actually get there, and it's a case of what can we do within the work that we're already doing to trial the approaches, to work out what that means for our data going forwards in terms of being able to compare to the work from the past. Because as Henk said, there's a lot of kind of step changes coming along, but particularly if we're talking about anything to do with monitoring and looking to assess change, what we need to understand is, well, what does that mean for looking back at the older data as well? And all tied up within that, we had quite a lot of discussion and conversation going on, which is really needing to be, again, open and honest and acknowledge that even with the best will in the world, any time that we create a slightly tweaked or a slightly new bunch of rules, be that additional new rules for the count and cover recording that we now are looking at undertaking or now are undertaking with annotation software platforms, and then looking one step forward, creating new rules for potentially cell count methodology. So that I call it cell count, cell frequency, all the same thing, the 5 or 10 by 10 grids. Wherever we look to start creating new rules there, we're going to have to be very open and honest, and again, working through the working groups is really useful to gather the feedback so that we find where those rules for the best intentions, or are set out for the best intentions, but still trip ourselves up. And I remember certainly in the working group, I think it was Alison came up with a couple of brilliant examples of situations where a simple rule that we've set out in the last couple of years for count and cover recording through annotation, it makes sense when we come up with it and when we write it down. And then in practice, there will always be a situation that comes along that makes it either really, really difficult, really time consuming, or just nonsensical. And work through the working groups to feed back on those and to try and see if there are solutions is going to be really, really important. And I think there I had one last bit on here. Yeah, and the last bit on here, I don't know, Jen, if you want to jump in, please do. But something we did go off on a slight tangent, but again, I think it's an extension of where we'd even envisaged originally this working group going, was looking at the real interest in the prospect of how we can incorporate within this quantification work, exploring the extraction of biomass data from imagery, which is something that certainly within the kind of SNCB statutory world, something that we just haven't been doing, haven't been really exploring. I mean, and within the academic side of things, there is work ongoing. There's some really interesting stuff going on, which, you know, if we can bring it in through the working group as another potential option in a toolbox, God, that sounds very cliche, but you know, that could be really, really exciting. So that there's stuff there that isn't even there in name in the tasks in the working group at the moment. But if things like that, we can build it into this decision tree, this list of tools, and set out where and when and how it's appropriate for it to be used, that would be a real win. Those were the points that I jotted down as my main kind of take homes from that working group. So as I say, what I haven't touched on, and what I know John was hoping to share, was, you know, just a few examples, mainly from, you know, specific projects or bits of work that are going on that are kind of going to come back and feed into this. But unfortunately, I don't have that. But what I'd like to do is leave plenty of time for Mark to come in with the with his bit on the actual annotation platforms themselves, because they are very, very key to all of this discussion, because without the annotation platforms enabling everything that I'm nattering on about, we're kind of dead in the water. But also, Jen, others who were at that working group, if there's anything you want to correct me on or jump in on, please do. But I'll stop there, otherwise I'll just be waffling.
Thank you, Ross. Thank you, Henk. Yeah, we've got time, so it's not an issue if there's something else that you needed to say. But I don't know, Jen, if you want to include anything more, or.
No, I think that was a great summary, Ross.
Okay, great. So we do have a question in the chat. I don't know if you want to touch any more on what Graham said about the SAC4 scale with the point data, or maybe that's best to discuss another time. He was talking about converting the point data into SAC4 and how that compares with the straight SAC4.
Yeah. I think it does take. Yes, I think that I agree, Graham. It does take us down a bit of a rabbit hole, and there's many on this call that are probably much better placed to go down that rabbit hole than I am. But it takes us down a rabbit hole with imagery and SAC4. There's a lot of mismatches in terms of how you apply it, because especially if we're talking about still images, because you've got such a small area that you're looking at. So backwards conversions, conversions between real count, SAC4, etc. Jen, save me, help me out here. But it gets really tricky, and
I mean, I think the main thing for all of these types of comparisons is, like, you need to set up a study in order to examine them, and you need to set it up properly. And it's extremely likely that we are just going to have to draw some lines in the sand and not worry about what we used to do. And some of those things we're just going to have to, you know, come face to face with. That, you know, the fact that we can't maybe necessarily compare our old previously collected data to our new collected data. Some things we may be able to sort out. I am part of a few studies that are currently ongoing, comparing different aspects of some of these things right now. So there may be some sort of academic research related to that might help inform us. But I think, like in terms of having an evidence base, there's quite a lot of things that you could compare. And so it's a bit of deciding which things are going to be the best things to work on first, which things are going to get us the furthest.
No, I completely agree, and I think that's where the last task specifically in this group really needs to link back with the main table that Henk put up before around the uses of imagery, because you very quickly can disappear down a rabbit hole of trying to unpick some of these things. But if we can just be really frank about what was the purpose, what is the purpose that we're trying to do now? Is that compatible? And if it is compatible, great, and let's do. And this is what we can do about it. But if it's not compatible, I think I would agree, Jen, that a line does need to be drawn. Otherwise, you never get out of that rabbit hole. There's a couple, Henk at least. I don't know who else has got hands up.
Yeah.
Sorry,
Sure.
I can't see any hands up right now. Sorry if anyone has theirs up.
So at the moment, so I think Henk was first, but then Graham and Mark, I've no idea what order you guys stuck hands up in, so we'll go alphabetically.
Well, I just a small thing, touching on what Jen said. I think it could be quite a neat target, and I mean, it sounds like you guys have really thought about this. Is your plan of conducting some sort of multi-metric study to compare the approaches, but set it up really well, getting the sample units nailed, which I think is the linchpin really. But is that going to be the focus of the group going forward, as a flagship study?
I don't think we've really talked about that, because, like, let's be honest, that would have many, many facets to it. The one thing that we haven't mentioned here that is obviously related to sample unit size is that the answer is different depending on what community you're looking at. Yep. So if you're looking at an area of just sea pens, that would be way different than if you're looking at some encrusting reef. It'll be way different if you're looking at a sponge garden. So I mean, you can see we're obviously getting to the same problem we had before when we were talking about data storage, which is like, this is an exponential growth situation of options here. So I think that's why prioritizing what things we can choose and work on will be the best. I might just respond to one of the comments that was in the chat by Neil Golding. I think that's a good point. The question is, if we're re-examining the Moore study, then it says, would it be good to flag the report with a note that the findings are being reconsidered? I would certainly take that off the done list. I would not put that as a task that's complete. And the question is really whether we can, for example, reanalyze the underlying data from the study in order to get at some of the questions, or whether we need to design some new aspect related to that and how that fits into the priority list. But yes, I would certainly flag that up. I would not use, because of sample unit size, and also some of the comparisons I don't think you can compare. So certain methods of annotation are for certain aims. They're not necessarily interchangeable. So you wouldn't compare them anyways, because if you had picked aim A, you would follow method B, versus if you picked aim C, you followed method D, and you wouldn't compare methods B and D because they're not for the same purpose. So that should be part of our decision tree that's developed as part of this group, is considering those things. Sorry for so much waffle. This is one of my primary areas of research.
No, it's great. It's much appreciated, Jen. I think Henk, Mark, be primed and ready to go in 1 second. I'll shush. Henk, just going back to your question, certainly it is something John and I have discussed, kind of a bit of a pipe dream and an ideal situation of, again, looking at the different people we've got engaged through the working group and the data sources and the access they've got to so many different types, and as Jen was saying, data collected for subtly different purposes, and that would that you would apply different approaches. There's definitely the opportunity there for a wonderful experimental study. The crux of it is who's got the time and the resource to go into that. So then it's a case of working backwards and looking at, for us anyway, looking at what aspects of that we can build into the work that we're already doing, so that we might not be able to answer all the questions in one go, but if we can start feeding in small bits of the questions in through the working group, we can try and bring that together. So I think that's the answer to your question. Is I would love to say yes, there's a holy grail and we've got it all here ready to go, but it's going to be piecemeal.
Yes, it's
Mark,
a design question as well as a funding question by the sound of it. Yeah.
Mark, do you want to jump in?
Yeah, I actually think Jen hit the nail on the head when she said that it depends what you're looking at as to what method you're going to use. And we are not going to come up with one method that is the best. It just isn't. That isn't even worth approaching. I don't think it's worth spending any money on.
But I don't think
Especially
that's the plan.
Yeah, well,
I don't think that's what we're trying to do. I think what you want is a decision tree to help people to make the right decision for their project, rather than telling them what the decision is.
as in pros and cons of each approach.
Well,
Unless the plan is to try and get everybody's data comparable, but I don't see that as, you know, not without doing a lot of standardization. I don't think that's
Too many different objectives.
Too many different objectives and not approachable. But one thing that really stuck out in the meeting to me was when they were talking about biomass was, well, instead of putting a point on a data to count it or to put it in a cell, why not draw a line or two lines or a polygon? In which case you might not be interested in biomass, but by doing that, somebody else down the line would be able to come up with a proxy for biomass. And that sort of information, that sort of recommendation is really useful. That really stuck home with me. I then looked at it and thought, you know what? It doesn't take me any longer to draw a line or a polygon. And if that can then be useful further down the line or give me more options or someone else more options, that's a really valuable bit of information.
No,
I think that's a good point, that you need to consider the other uses. And to that end, I think that we can probably constrain this myriad of communities and purposes, aims, objectives to a reasonably short list that are related to conservation purposes. So conservation and conservation related studies. I think we can probably narrow that down to not a huge number of options in order to kind of guide people into doing things that are at least at some scale quite comparable, as well as meeting the purposes of their project. I think it's possible. I'm optimistic.
Okay, great. I think, Mark, your hand is still up. If my hand function is still working. I'm not sure if there's any other hands up at the moment.
Graham's is still up. I'm not sure.
Oh, is it?
I don't know if that's been picked up. No, it doesn't look like he has got.
No, I did. Yeah.
Sorry, Graham.
It was well, a similar question, similar point to Mark actually. That with the, I suppose, the added value of some of the sort of the more granular methods of counting, sort of, you know, with the whole machine learning stuff before. But added on to that, obviously with the original report looking at, you know, quantification methods, I believe it was just raw count. It was just a count per image that was provided, rather than in effect annotations that were done on the image. And so there's sort of two aspects of that, just with the whole, you know, the fact that it's really moving towards, you know, annotation tools and the speeds that you can annotate images now, especially if you're using things like tablets and styluses. You know, your eyes are going to have to see the objects on the screen anyway, so you can just tap. I suppose taking raw annotation into account in the next iteration of this sort of study, to actually see, you know, just how quick or slow is it, and in effect, it is to me the one that can basically, from raw annotation, you can create any of these quantification methods. So whether that's the possible route forward.
Okay, great. Thanks, Graham. Just been a comment from John in the chat. I don't know if anyone's seen that. He suggests that the reanalysis of the Moore report is not worthwhile. saying there's a key flaw of the design. There was no attempt to start with a standardized list of taxa or at least criteria for adding qualifiers. He says that this level of inconsistency in the data was massive. And Jen was saying that there's some useful things we can still do with it. Anyone else wants to comment on that, Jen or Rosser?
I'm not close enough to it.
Okay. Guess we can leave it at that unless someone else wants to jump in.
I could just say, I mean, there's a lot. Well, there's a story that could be written about the story of the report. As John and I know, and Alison and everyone who worked on it, and I think it's safe to say that we've learned a lot since then. We've learned a lot, and the take-home message is that we're still learning, and it's great that we're all working together to try and make it better. So, yeah, I guess to take the best lessons forward and to learn from them is probably the best thing that we can do.
Just quickly, Jamie Davis has got a hand up, Steph. So Jamie, if you want to jump in before we move on.
Yeah,
Thanks,
sorry,
Ross.
I'm having a bit of trouble seeing all the hands. I just saw it now, so thank you.
Just a quick question. So I think the idea of what was done for the report is good because we need to know the limitations of different methods. I'm surprised the SAC4 didn't do a little bit better, actually. Did you use a SAC4 macro so everyone was using the same size range for the taxa?
Yes, we'd have to pass it on to John and Alison, or either or. Yeah, sorry, go ahead.
There was a standard SAC4 scale being used, but yeah, how well that was applied, and we all know how difficult it can be to apply SAC4 consistently anyway.
Yeah, what I mean is you kind of decide the SAC4 size range almost before you do, as you're making your species catalogue almost, so everyone uses the same size range for different, for the same taxa, if that makes sense. It makes the conversion from the absolute numbers to the SAC4 a lot more consistent. So I wonder if that would have helped a little bit with those numbers improving, possibly.
Possibly. There was, scales were supposed to have been applied, but more attempt to standardize that, and I think the primary thing we should have been doing at the beginning was to do a small study initially to get a list of the species that we expected to see that were there. If we'd started a list like that and decided which species would go with different SAC4 scales, then we could have cut out a lot of the issues of the data set.
Yeah, that's what we tend to do. We comb our imagery first off, get our species lists together, then do the create the SAC4 macro based on that, and it just makes it a lot more accurate in that sense. That's why I'm surprised the SAC4 didn't do better, because in theory the SAC4 should have less noise in it because it's not using absolute numbers like abundance. So yeah, that would probably help it.
Well, I've got a lot of experience of using SAC4 and trying to get people to apply it consistently.
Really?
I've done that over many years, and I'm extremely frustrated with those scales.
Yeah, it's difficult, isn't it? We tended to do it as a post-processing thing afterwards, where we built a macro in Excel and we did it that way. So people just did their absolute, the abundance, and then we would convert it afterwards. But I know what you mean. It's not easy to use as a.
Okay, right. Well, this is going to be quite quick because I haven't done anything. I'm not sure I did volunteer for this job. I think I said I was vaguely interested. But anyway, I've got this job for the Image Annotation Software Group. I haven't had much time at all in the last 2 months. And to be honest, I'm not going to have much time until about another month's time. But James King has been helping me. And I'm going to use this talk really. I was going to apologize for not doing anything, but now I've listened to a lot of people in this workshop. I think it's quite a good thing I haven't done anything, because I can use this to try and work out what we need to do with this group, because it hangs on so many of the other groups and what they're doing as to what we come up with. So there's Henk's picture. Back in 2016, there was a very good paper which sort of went through a lot of the image annotation software that was available for marine images, stills and video back then. But 2016 is a long time ago, and to be honest, 3-4 months is a long time in this particular field. James went through on a little search for me a couple of days ago, and I guess it depends how long you spend, but this is sort of the list of current amount of marine image annotation software he could find, all very variable. Back in 2016, they also sort of quantified what it could do, what the various functions were. You'll find that's out of date as well. I used Biggle on a project about 4 months ago. I went back into Biggle last week and it's changed again, particularly on the machine learning side of things. So things in this area are developing all the time. We've got some of the old standard ones with their classic structures like Squiddle. I haven't used that personally, but the Katami structure there is very suited really to the southern hemisphere and the more sort of subtropical things, but could easily be adapted, as the EIP could be adapted to take that in, and the ID Approaches group could work on that. We've got video-based ones. VARS seems quite hard to use outside of the actual establishment that actually set it up. A new one, which I thought was quite interesting, it was in a recent paper I had a read of, was CoralNet. That seems to really take machine learning to its heart and starts actually learning itself, as far as I could read. I haven't used that one itself, but it looks like it starts actually analyzing the images and doing a sort of machine learning thing in the background, and will actually start, as you come onto your images, once you've got enough in there, it will start making suggestions as to what you should be annotating things. So obviously there's some sort of machine learning bit going on in the background there. There's real-time stuff as well. So if you're doing video surveys, there's all sorts of stuff now where as you do the surveys, you can annotate them or put notes in, which might be quite useful. Probably not that useful for actually doing the data extraction, but maybe will make the data extraction a lot more useful and quantifiable afterwards. But these things are all available. This one looks great. I like the banner on this one more than anything, and the strap line at the top. It seems to be point-based though. Now the difference with this one is you download it and you work on your machine with it, whereas a lot of the other ones involve putting images into an off-site store and pointing a web-based application at those images. This one does allow you to work on your own machine with your own images in standalone capacity. However, it does seem just point-based, and if we go back to Jen's point that biomass might be really important, I don't think this will be that useful, because I don't think you can, I haven't used it in anger yet, but I don't think you can actually draw polygons. So that might be a case where this particular software looks great, but maybe won't meet our needs when the quantification group sort of come up with their findings. I've used a similar, if I'm actually doing stuff at work, I use something called Image which is a really bog standard piece of medical software, I think originally not really designed for the marine field at all. And Image though is literally you have a notepad next to you and you go and count things or draw things. You can export some of the data, but it's certainly nowhere near as advanced as something like Biggle, however you pronounce it. This is one I have used a lot with a student. Now, I guess depending on your organizational setup, I really struggled to get NRW to allow me to set up a web-based folder with complete open access that I could point Biggle to. In fact, the only way I got to do this project was to involve a university student from Swansea University, Jess. She set up the folder. She put in a request, a data request to NRW to have the images. She was then able to take the images and load them onto this open source folder where Biggle could then look at them. So that's a restriction for me. using this day to day in my own organization. However, students are great, and getting students to do it is always good in my book. And once I got into BIGL, I did a lot of the features that are in these web-based annotation tools are really, really useful when you're running a project. So I didn't actually have to do a lot of annotation here. I certainly had to play with it to begin with. And Jess and I ran through building up our own label tree. So on the side here, you can see a list of things that can be taxonomic, morphological, or a combination of the two. I really like the way I could bring in the Katami structure from CFAS had made their Katami structure open source. So I could bring that in and take bits from that. You can link with worms. And once you've created your own label tree structure, you can share that with other people. You can see how, if we're working in this sort of group, these sort of things are really, really useful. You can also do points, lines, polygons, that's all there. And you will get an area when you do your data dump at the end of it all. We certainly learned a lot, and I tended to go for more polygons than points in the end. Just even drawing a small circular polygon, I think, particularly, and now we're talking about the potentials of biomass. I think that has some real legs to go with it, and avoid points. And even, I mean, you do get a count as well. So you get an area for the polygon and a count of polygons. So even all these are small circles. I'll get a count of the circles. So I've still got the count, but you've also got an area as well. And that just opens up other options. Personally, from my point of view, running a project with a student, I really found the BIGL's QA tools really, really useful. The label tree meant that through group working with other colleagues in NRW and with the student, we could come up with a defined list of how we were going to record stuff. So just like John Moore's mentioned there, that's a big improvement. If you want to do any of these types of studies, is to actually go through your images first, come up with a species list of what you're likely to see, and define how you're going to call those things and to what sort of morphological or taxonomic aggregation you're going to use on those sort of things. We did decide to measure as well as count, so small polygons, things like that. The QA features available in BIGL as a project, if you like, running the project with a student, was really useful. They have something called the Largo tool where you can bring up, so these are all the Caryophilia smithii images, and there's a whole stack of them, and it brings them all up as annotations. So rather than just labels, these are little segments of the image with a bit of context behind them as well. So the actual annotation would have just been a small circle around the size of the cup coral itself, but the Largo tool brings up some contextual information around the side, and you can just run through these, looking for sore thumbs sticking out. Has these all been consistently labeled? Is there anything weird in there? Actually, you can bring up each individual classification or annotation, and you can just check that, do we need to change all of them? You can look for odd ones out, or you can say, oh, we've called the whole thing wrong, and change the whole thing. So that was very useful as well. And there are machine learning features in BIGL, and I would have really liked to try those out. Unfortunately, the student ran out of time, and my second student got a far better offer in Malta, so I lost her, unfortunately. But I would really like to try these out. However, I do realise it isn't the golden bullet I thought it was going to be. And that's just my naivety of what can be achieved here. As has been said before today, this is teaching a program to look at one set of images. The lighting, the background situation, all have to be fairly consistent for it to learn what a particular annotation is. The annotation tool in BIGL is particularly designed not to tell you what something is, but whether something is worth annotating or not. Does it stick out from the background? That's its primary purpose. You then go in and annotate it, but it does the background job of just picking out what is just background rubbish and what is something living that's worth you giving a name to. There is a way of training it, and perhaps cup corals would be a good way of doing this, actually, is actually giving it a consistent load of background images with cup corals in, and then continually killing it just to look for a cup coral. However, even if you did that and you trained it up on your thousands and thousands of cup corals would be needed to train it. It would only work for that data set. And the chances of transferring that knowledge, which also isn't very easy, as people in that field will tell you, actually transferring the learning is very difficult. But even if you then applied it to a different data set with different background or habitat conditions and different lighting, it's very unlikely to work. So I think we're a long way off here, but if we're going to get it to work, we need to be doing all the things like the EIP and the quantification stuff and feeding into the machine learning side of things so that we've got lots and lots of annotations of labeled species in a whole wide range of backgrounds, and then possibly we can sort of link all this together. Really then, this is my main aim of the talk today. Is I haven't had any meetings like the other people have, so I would just like to make sure, well, this is my main question. What really do we do? Because to me, it all hangs on what comes out. The EIP absolutely underpins this. And I don't want to prescribe to people what annotation systems they should use, because there's so many out there and they've probably all got plus and minus points, and it just depends what you happen to be researching. So I don't want to be prescriptive in that way, but possibly we could give a list of desirable features that an annotation tool should have. And that might be, how does it output data? Can it just do counts? Can it also do areas? Can you just draw lines and points, or do you need polygons as well? We really need to, these list of desirable features, however, will come from the EIP group, the data group, the machine learning group, and the quantification group as well. And my question really today is what you think this group should do. We could just keep up to date with developments and act almost like a repository of information where we can disseminate information out to the wider group if new bits of software come online or new bits of recommendation come out of some of these other groups that mean that certain tools are more suited than others. I don't know. I mean, I guess we could go all the way down the line of producing a similar paper to the 2016 paper. However, I'd be very aware that that's probably would be out of date within a year. So whether it'd be worth writing an entire paper on it or not, I'm not sure. That's it for me. I haven't got a lot more to tell you, so I've got a lot of questions really, and really I'd like to open it up and think, what should the annotation group do? I think it's a lot clearer what the EIP group, the data group, the machine learning group, and the quantification group could do, but how do we then, as an annotation group, and whoever wants to be involved in that, what's our role?
Thanks for your talk, Mark. Not sure if anyone, there's quite a bit going on in the chat, but oh, Ross, you have your hand up. It's the only one I can see at the moment, so.
That's cool. I think there was myself. I saw Jen's hand popped up very briefly afterwards, and Graham. So there's a couple of us. It was a very quick plug, actually. Mark, you picked up on something partway through there, and it completely slipped my mind to mention it in the quantification group bit. And I don't know if Joey mentioned it in the identification bit earlier on, because I was still trying to make some lunch. Mark mentioned the roughly vaguely Katami-based label tree that we came up with in CFAS 18 months ago or so, which is after the last big picture meeting. As Mark said, that is out there, that is available, and the absolute intention with that is to keep using it, not just ourselves, but to be using it as, you know, within this community, anyone that wants to really get involved, and to really feed it into and feed off things like the EIP work and the work of the working groups to keep developing that. And it was a big, the whole Katami thing for the UK was a big discussion in the, I now forget which working group it was. It was either in the quantification group or it was in the identification group. They all blur together. But yeah, it was just a bit of a plug for that, because I forgot to plug it earlier, just to make people aware that that is out there. It was in the paper that Robin was mentioning in the chat earlier on with the freshwater lens work. Those guys were using it, so it is out there and it is available. And if anyone who's interested in getting hold of it and working with it and working with us to develop it as a collective, as you know, it fits somewhere between the annotation group and the identification group and the quantification group. We're really keen to keep pushing that and keep that moving on. That's not intended at all to be kind of a static product. That's got to be a living thing that we develop collectively. Anyway, sorry, I'll shush. Crikey, there's a whole load of hands.
Okay, thanks, Ross. Yeah, sorry, I'm still having a bit of trouble with the hands. I can only see Graham's up. I'm not sure who else.
I think Jen was next, and then it was somewhere between Henk and Graham.
Okay, sorry, Jen. You can go next.
Oh, thanks. I think this group has opportunity to be active and not passive in the development of these annotation platforms. I think that's probably the biggest opportunity here. Just as we can alter the course of our destiny in other areas, we can here too. And so we should. And as I mentioned yesterday, all the people who work on these platforms are really approachable and want people to use them. So they want to know about improvements and suggestions. So I think that is probably the biggest thing that this group could do. I have worked with the developers of Vars and Squiddle and Paparazzi and Beagle, and all found them to be really engaged and interesting to hear what users have to say. So I think that would be the biggest contribution. And so maybe there's different ways. We talked yesterday about how that might fit with some of the things from the EIP or those new spreadsheet pro formas for outputs and things like that. But it certainly should go beyond all those types of things. I think the second biggest thing that this group could do is to get everyone using a data-based annotation system. As you mentioned, just the opportunity to easily QA/QC your data, your identifications anyways, makes that a huge step. So even if those were the two things that this group did, I think it would be immensely valuable.
Okay, great. Thanks, Jen. Graham, do you want to go next?
There you go. You missed me saying I'm going to put my very boring standards hat on. So going to talk about
are really approachable